This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 172, recorded on February 24th, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from the Department of Microbiology and Immunology at the University of Michigan Medical School is Kathy Spindler.
Hi, Vincent.
How you doing?
I'm doing okay.
It's a second time for you on TWiV, isn't it?
It is.
We were out there. We were out there in November, Rich and I.
November, mid-November.
I bet it was colder then.
It probably was.
I remember it was freezing because I wore a light jacket and I almost died. And I wanted to walk around to go see that building where they had announced the polio vaccine results.
Oh, right. The Rackham Graduate School building.
Rackham. Mm-hmm. And I froze because it was early in the morning. So today here it is 4 degrees in New York. It is cloudy and raining. Horrible day.
So we've had some snow off and on, or rain, or spitting or something, and it's 34 degrees Fahrenheit and 1 degree Celsius. And that reminds me that I've wanted to tell you for a long time that someone corrected me when I was talking about the temperature in centigrade. Mm-hmm. And evidently there are numerous temperature scales that are centigrade, but the one that we use is more properly called Celsius.
Mm-hmm. It's not the same, huh?
So I guess centigrade is more generic. So Celsius is specific.
What does it mean?
Well, it's named after this person, Celsius. And
yeah, Andrew Celsius, a professor of astronomy.
So anyway.
So we should call it Celsius? That would be more accurate?
Right, right. And now evidently it's been re-standardized to, with modern things that they can standardize it to. But anyway.
Yeah, I think we should, as scientists, always use Celsius. Mm-hmm. I have it in my car on the dash, and that drives everyone crazy because they don't know what it means. But they should learn.
Right. Well, and that reminds me of something else, which is a friend of mine in England told me a couple more temperatures that you can all keep in mind, sort of a little mnemonic. So if you just remember that 28 is 82 and 16 is 61, you can often then interpolate a lot more of, outdoor temperatures. So that's 28 Celsius is 82 degrees Fahrenheit, and 16 Celsius is 61 Fahrenheit.
Wow, 28 is 82. The only one I remember is 20.
Yeah.
Which is 68, right?
Right. And 37, you probably do.
Is that 98.6?
Yep. Yep.
Wow. That's why we keep our incubators at 37, huh?
Right. And that way, you don't have to remember, is it multiply by 5/9 or 9/5 or add or subtract 32? You can just do some interpolation.
The alternative is just to get used to what each temperature feels like, right?
Right.
I mean, that's what works for Fahrenheit, because you know that 90 is really hot.
Right.
And so for Celsius, you'd have to know 37 is really hot.
Right.
Or even
exactly.
32, right?
Yeah.
Interesting. Well, 4 degrees, lousy weather, and you got snow, you said?
Well, this is the kind of snow now that's not sticking to the streets or sidewalks because the ground temperature is too warm and the air temperature is too warm.
Cool. All right.
So, yeah.
Well, before we move into science today, I just wanted to make a note of a comment on the blog. I think it was at twiv.tv. After TWiV 171, which was called 1 is the Loneliest Number. Mm-hmm. So I put a link to 3 Dog Night in the show notes, and I just wondered if anyone would get it. I don't know how old our audience is. I guess it's a range.
Yeah.
Anyway, someone posted, it's actually Ed from South Africa, who's a virologist. You may know him, right?
I don't know.
No?
I don't think so.
He said he wrote any virology blog that includes a 3 Dog Night link is a blog for me.
Well, that's good.
That's a good one. Okay, today, Kathy and I, and as you can probably figure out, it's just the two of us. It's been a long time since we had a duo on TWiV.
Okay.
Do you know offhand? Probably.
No.
You know,
I know I've heard some, but...
I think it's 138.
Okay.
It was Raul Andino and myself.
Mm-hmm.
I went out there, and when I travel, I like to bring my recorder and try and do one-on-one TWiVs. So I think that was the last one. All right, today what we have in store for you, we have a paper and a discussion. And we'll keep you in suspense for the discussion. The paper is one you picked.
Yeah. When we do journal clubs, we tend to describe first why we picked the paper. And if I were doing this one for a journal club, I would have had a number of reasons. First of all, the science information, the content really grabbed me. So the title is CNS Recruitment of CD8 positive T lymphocytes specific for a peripheral virus infection triggers neuropathogenesis during polymicrobial challenge. So that's a really long title, but hopefully we'll be able to explain it. And the authors are Christine Matullo, Kevin O'Regan, Mark Curtis, and Glenn Rawl from the Fox Chase Cancer Center. And so, as I said, what attracted me is this idea of looking at what's going on during a polymicrobial infection. And
why do you think they use that term polymicrobial? Isn't it polyviral?
Well, polymicrobial is, I think, the standard term in the field. So that's what I've seen in other papers.
Do you think a virus is a microbe?
I do. I do think of that.
Some people don't.
Well, whenever we have to fill out little surveys and say, what kind of department you're from, or what's your area of research, it's seldom that they list virology, and so we have to pick microbiology. Yeah. So
we were both in microbiology departments.
Right.
But I was just reading a book called Microbe by Elio Schechter and a few others. In the beginning, they define microbes, and they don't include viruses.
Oh.
Even though there is a virus chapter in the book.
Okay.
Sorry for that diversion, but I was just curious about that. I thought it might be because they didn't want to have virus twice in the title. Virus infection and polyviral challenge.
Yeah.
So, Glenn, did you do this for a journal club?
No, I haven't. No.
But you would if you were going to do one, right?
Sure. And I'm not scheduled in this year's journal club list, so this is my alternative, I guess.
This is better than a journal club.
Oh, I think so.
More people hear this one.
Right. So, and I guess Glenn has been on before.
He has.
I looked it up. Was it 38, TWiV 38, talking about measles and other topics. So the other reason I like this is I think it's just a really well-written paper. And so I thought about Sophie, the listener from a couple weeks ago, who wanted help on how to read a scientific paper. And so it's almost a spoiler alert. I think you could turn off the TWiV now and see if you could read the paper. And because it's the kind that takes you through all the figures, and when they make a statement, they direct you to what part of the figure it is, and it's open access, so everyone can take a look at it. And things in the conclusion, the discussion section talk about what they've found, and also where they're going, how they can find out what they're doing to try and figure out a mechanism. I just thought it had all the components of a good paper.
Did you like that paper discussion we did?
Yes, very much. I sent it out to people in our department and former students of mine that teach at various types of colleges and universities, and got a lot of positive feedback from that.
I broke it out for you separately.
Yep.
So if anyone who's wondering, was that TWiV? I have to look. Let's go to the website. Episodes, what was that called? That was the epidemiology one, 169.
Right.
We did a discussion on how to read a science paper, and the whole episode is 2.5 hours, so I broke it out.
Right.
I think it's a half hour on its own. I was amazed, actually, how long that went.
Right. But it was intriguing, all of it.
It's obviously something that everyone's passionate about.
Yep.
So check that out if you haven't listened to it. Yep. Glenn was on, and he talked about measles. I should have him back on again, but he has a terrible air noise in his office.
Oh, dear.
Very, very bad. And unless he's moved, it'll be tough. It's a bad reason not to have someone on, but there you go.
You could maybe do it from home or find someplace else to do it from. Yeah.
And make him stay home to do TWiV, right?
Right.
So what's the basic idea here?
Okay, so they have, as they state in the abstract, they have the hypothesis that viruses don't have to replicate in the tissue in which they cause disease. And so they use 2 viruses, and figure 1A really kind of shows the whole picture of the paper, kind of in a graphical abstract. So they use 2 viruses in which they can restrict the viral replication to a certain part of the animal, in this case a mouse, and then they determine that having replication in one part of the body of one virus affects the outcome of disease and actually results in significantly more mortality in the other part of the body where the other virus is.
Let me ask you, why would you have this hypothesis to begin with? Why would you think that a virus would cause damage where it's not replicating?
Well, I think that particularly with respect to the brain, there are a lot of diseases for which viral etiologies have been proposed, but no virus has been identified. Mm-hmm. So, in fact, in their discussion, they specify a couple such diseases. And so the idea is, well, maybe a virus is participating in some way and making things worse by replicating elsewhere. And I think they show that in this paper, the system.
Right.
So, so to be more specific, they use 2 viruses. They use Measles virus, which doesn't replicate in mice ordinarily, but they know several of the receptors for measles, and so they have a transgenic mice in which the human CD46 molecule, which is one of the receptors, is expressed only in the brain. And so if they infect mice by putting the virus directly into the brain, into these transgenic mice, they can get a viral infection, but they find that 100% of the mice survive. And then the second virus that they use is one that we abbreviate LCMV. It stands for lymphocytic choriomeningitis virus. So you can see why we abbreviated LCMV. And this virus has been used a lot in viral immunology because you can get all different kinds of disease consequences depending on what virus you use, how old the mice are, if they're immunocompetent, and various other viral parameters. So they use a condition where they infect adult mice, I think it's adult mice, in the peritoneum. So they introduce it intraperitoneally, and LCMV then is restricted to replicating in the peritoneum.
It's interesting that you could put measles in the brain and they recover.
Yeah, 100% survival. And then again, LCMV in this peritoneal infection, 100% survival. Mm-hmm. So then, as pictured in this figure 1A, you do the dual infection at the same time, and that's kind of a key point, as they show a little bit later. If you infect them concurrently with both viruses, you get 50% mortality. So that's really a striking finding, and it's showing that having these 2 different viruses in this polymicrobial infection is changing the outcome. And so this is really a viral immunology paper, a viral pathogenesis paper. So for those that haven't had much immunology, hang on, we'll try and take you through it. But what they find is that recruitment of CD8 positive cells that are specific for the virus replicating in the peritoneum, in the body of the animal, those CD8 positive T cells get recruited to the brain and make the brain pathogenesis much worse, and that leads to the death of the mice.
Just half of them, though, not all of them.
Right, not all of them. That's true.
Okay, and then so this figure 1 is one way of measuring besides death, right?
Right.
Which
so
is
in
weight loss. It's a common thing to do with infections, right?
Mm-hmm. And interestingly, they they do what we call stratify the data. They infected mice, or what they call doubly infected mice with the two viruses, and what they found was that 50% of the mice roughly would lose a large fraction of their body weight, and 50% of them had mild illness but survived. And so they, in several of the subsequent figures, they again stratified the data and look at the results for the mice that had the severe illness and the mice that had the mild illness.
And the uninfected mice and the singly infected don't lose any weight.
Right.
They're okay.
Right.
So do you think that they initially put all the data together and they found there was something weird and that's why they stratified them?
Could be. Or if you pooled all the data, you'd probably get an effect that wasn't quite as striking as when you pull out the severely affected ones. Right.
Maybe someone was looking at the raw data and said, hey, some of these are losing more weight than others. Let's separate them.
Right.
Sometimes how science works, right?
Right. Mm-hmm. Definitely. Right. So then when these guys die, I guess you're going to tell us how they die or what the symptoms are. They just drop over or something happens? Let's see. Well, okay, so it's different. When they die from the measles virus, well, in fact, they don't die from the measles virus alone, but they show, well, let's see. In figure 5b, they do show the posture that the measles virus alone gives the mice, and that's a sort of a hunched posture, which is what I think of as a typical sick mouse posture.
Yeah, they call it overt signs of CNS disease, including ataxia.
Right.
What's ataxia, Kathy?
Yeah, I always have to look that up.
I think it's
something about moving, right?
It's uncoordinated walking, I think.
That's right, yeah, I think.
Let's see. Ataxia is a form of cerebral palsy, wobbliness. There you go.
Right. Okay, loss of full control of bodily movements.
And tremors, a hunched and ruffled appearance. Now, here's a word I didn't know. Kyphotic.
I didn't know that one either. I meant to look it up.
Kyphotic. Hey, Glenn, what are you up to here? You could just say a hunched and ruffled appearance. And then they have seizures and die. That's very interesting.
Right.
10-second seizures, and then death is always coincident with the seizures. So those are the doubly infected mice. They die.
Okay.
They show those symptoms, and then they die. And those are the severely sick ones. And, of course, the other half, they don't lose weight and they don't die either.
Right. But the doubly infected ones, or if you put LCMV directly into the brain, you get this weird posture. Instead of the hunched posture, you get this decerebrate posture in which the limbs are held straight out and the toes are pointed down and the head and neck are arched back. So quite a different look from the hunched posture. So the doubly infected mice, where the LCMV was put into the peritoneum, end up having a phenotype, posture phenotype that's like LCMV going directly into the brain. So.
Right. Now, they also ask whether you need virus replication for this.
Right. So this is in Table 1. They treat virus with UV, ultraviolet light, which would keep the virus from replicating. And they do it for each virus individually. And if they UV treat the measles virus, but they still do the double infection, then they get 0% morbidity. So that says that the measles virus has to be able to replicate.
Right, and that's in the brain, of course.
Right. But if they do the double infection and they UV treat the LCMV virus that's going into the peritoneum, they still get morbidity, although not quite as high as with the untreated LCMV, but still a significant amount of morbidity. Mm-hmm. So that says that the non-replicating LCMV doesn't have to be present either in the CNS or, in this case, elsewhere in the body. Mm-hmm. So to get this phenotype of the strange posture and ataxia and all those things you mentioned, you don't even have to have a replicating LCMV.
You just need antigen, I presume.
Right. So that pretty much takes us through
that table also has the interval that you talked about earlier. If you put the two viruses 7 days apart, nobody dies.
Right.
7 or 30 days.
Right.
And I suppose if you went shorter, you start getting intermediate phenotypes. So probably a day or 2 days. It wouldn't matter that much, but it's clearly they have to do something together. It's interesting.
Right.
All right.
So I just remembered another thing I liked about this paper.
Yeah.
Although it's a PLOS pathogens paper, and they would have the ability to put all the supplemental data in that they could possibly want, they do things sort of the more, what I would call old-fashioned or traditional way of putting a lot of things as data not shown. And they seem like reasonable findings, and much of the time it's negative data, and so they just say data not shown. So, for instance, they say it doesn't matter whether they use mice 3 months old or 6 months old, and it doesn't matter whether they use female or male mice, and those data aren't shown. And so,
what do you think about that? Would you want to see it? Probably not, right?
No, because it seems logical and believable, and I take their word for it. So,
do you think that this is what supplementary data often are? Things that would normally not be shown, but they're requested, or the authors decide, well, since we can put supplementary data, we might as well put everything in?
I think so. What do you think?
I've never published supplementary data.
Yeah.
I just don't think, I don't find it useful. I think it should either be in the paper or data not shown.
Right.
I don't see the point of it, because if you're online, like this journal is, you could just put it in the paper, right?
Right.
I just find when you're reading a paper, and sometimes the sup data you want to see, and it's just a bother to have a separate file or somewhere else to go.
Yeah.
I don't like it. Maybe because I'm an old guy and I'm stuck in my ways, but I don't find it particularly compelling. I often read papers without looking at the sup data. It's almost like data not shown.
Right.
I don't have to see it.
Right.
So.
Yeah. We published one paper once, but it was a list of a whole bunch of primers that we used for a genotyping analysis. So it was a table of, and we put it in Journal of Virology, which ordinarily won't do that except for large data sets. So they consider that a set. So it would have been an extra page and increased the page charges.
Yeah, right. You don't want to do that. No, if that's the case, you put it sup data for sure. You don't pay for supplementary data, right?
No. I don't think so anyway. Yeah. So maybe that's why people do it, to get lower page charges.
I think in PLOS you have page charges too, right?
I don't know.
I think there are. That's the model for supporting the journal, I think.
Okay.
To charge you for it. Anyway, back to this paper.
Right.
So now the question is why, right? Why are these mice dying of neurological disease?
Right. Well, first, though, people might be really worried about, well, how good is this restriction of replication in the brain and replication in the spleen or the liver or where else? So they look at that in Figure 2 by a couple of different ways, and also they tell us about a piece of data that's not shown. So they look at, let's see, they're doing RT-PCR for measles virus from the brain, and BD, what does that stand for? Oh, below detection. And they see below detection levels for the LCMV in the brain. And then in the spleen, they have below detection limit levels of measles virus, but they have substantial levels of LCMV, which is what you expect, and the same kind of thing in the liver. And then they also say that they looked by immunohistochemistry, and they don't see any evidence of LCMV in the brain. But, of course, that's a negative result, and something like this is maybe a little more compelling.
So I suppose if LCMV had gone into the brain, that could explain the neuropathogenesis, right?
Right.
You could say that, well, when you infect with measles, it allows LCMV into the brain for whatever reason, but that's not the case here.
Right. Mm-hmm. And in fact, in the second part of the figure, they look at virus load, and that's also consistent with measles only being in the brain. so on. And that it's the same when they have the dual infection. It's pretty much the same virus levels. So
their conclusion from that little section, this was one of the things I liked when I was describing how I read a paper. Some people make a little conclusion at the end of each section. So the name of this section that we've just discussed, Figure 2, illness in co-infected mice is not attributable to mouse characteristics or changes in viral tropism. So that's the effect of age or looking for a virus where it shouldn't be. And at the end of this section, they say these data indicate that key aspects of the viral life cycle for both measles and LCMV, including tropism and load, were not changed upon co-infection.
Right.
So you don't even have to read the whole section. You can get the end result. Look at the figures and get the end result if you want to go through this quickly.
So then the next thing they do is start to look at infiltration of inflammatory cells into the brain of these co-infected mice. So title of the next section is Massive Infiltration of CD8 positive T cells into brains of co-infected mice. So this is figure 3, and they, at the top part of the figure, 3A, they show us in the first 2 panels on the left that there's no evidence of LCMV in the brain of these LCMV infected mice when they get only the LCMV virus. So intraperitoneal LCMV, they don't see any evidence of CD4 or CD8 positive T cells in the brain because that's showing the restriction again.
So this is a section of brain which is stained with antibodies to detect these particular populations, right?
Right.
Okay.
And then the middle panel, they're looking at just the measles virus infection, and they show a substantial and equivalent amount of CD4 and CD8 positive T cells in the measles virus intracerebrally infected mice. And then the key thing is then the third panel, pair of panels, where they have the dual infection, and they don't see much change in CD4 compared to just the measles virus alone, but they see a big increase in the amount of positive staining for CD8 cells in the dual infected panel. And then they go on to quantify that in the next part of the figure. They take brains from just the measles virus infected mice or the dually infected mice, homogenized them, and counted them by hemocytometer and then also by flow cytometry, labeling the specific types of cells, the CD4 cells or the CD8 T lymphocytes. And they see that there's an increased number and proportion of the T cells in the doubly infected mice. And this is one of the places where they then stratify, again, their data in the bottom part of 1B into the mice that had the mild or the severe disease. And there was no significant difference in the percentage or total number of CD8 cells between the mice that had the mild or severe disease, which I found interesting. So the lethal disease in the severe group compared to the mild group is not just due to the abundance of these infiltrating cells, because they have the same amount of cells, and yet they have a different outcome.
So does this mean that an increased infiltration of CD8 positive T cells causes the neuropathogenesis?
Well, that's what they're going to try and show in a few subsequent figures, and it seems to at least correlate with it. I don't think that they can say it's causative at this point.
Yeah, I just want to make that point because so far we have an association, right?
Correct.
You have to do some kind of experiment to actually prove that is what's causing the disease. You got to perturb it in some way, right?
Right. So they also do conclude that CD8 T cells are probably some kind of key player in the pathogenesis of these mice.
Right.
So then the next figure, figure 4, is the one that might ordinarily have have made me panic because, isn't this the one? Yeah, this is the one where they use what's called tetramer staining. Whenever I see tetramer staining, I think, oh no, I'm going to have to go look up some figure again. So I found a really good figure this time, and I think maybe we'll be able to put it in the show notes. It was just a Google image search, and it came up with the top hit was this beautiful figure, and it seems to be an open access article. But to make a long story short, tetramers are a reagent that allows you to mark the T cells that have a specificity to a particular epitope. So in this case, the epitope, or what kind of T cells you want to mark, are the ones that are specific for an LCMV viral protein. Mm-hmm. So one of the things you have to do to be able to make these tetramer reagents is you have to know the specific epitopes, some sequences in the virus that are presented on specific haplotype MHC molecules. So people, for some viruses, have been able to do that and characterize the major epitopes. And
so these would be epitopes that are recognized by the T cells, right?
Right. So basically you make this fluorescent tetramer, and because it's a tetramer, you have more binding sites, and I think that helps increase the signal. And then you also can stain the cells separately for whether they're expressing CD8 or CD4 or something like that. Mm-hmm. So they use these reagents to try and look at the kinds of T cells that they have in the brains of the singly or doubly infected mice. So of course, in the uninfected mice, they don't see any LCMV specific T cells, and they use two epitopes. One's called GP33, one's called NP396. And so they don't see anything in the uninfected mice. And in the measles virus infected mice, the top right quadrant of these flow plots is where you would see a positive signal, and the measles virus infected mice brains show no positive CD8 positive T cell staining for these specific viral epitopes. And then in the key experimental part of the figure, the last column where they have the dually infected mice, they get 8% of the lymphocytes were specific for LCMV GP33, and 17% were specific for LCMV NP396.
Do you wonder why there is no LCMV control here?
Well, they could have put in an LCMV control, I suppose. But
I suppose it's already been well characterized, right?
Right, and we already saw in the figure 3A that there is no evidence of CD4 and CD8 cells, at least by IHC, in the brain. Yeah, I think it's probably been well characterized, so the reviewers didn't press them to have to show that. So, and they even say that this is likely an underestimate of the LCMV specific T cells because this is only looking at these two major epitopes. And then part B is really just a control to show that splenocytes from the LCMV infected mice can't kill or lyse measles virus infected cells. Mm-hmm. So that tells you that there's not cross-reactivity of the LCMV and measles virus epitopes in this assay.
So they say it's an underestimate, but would you think that all of the infiltrating CD8 cells are LCMV specific, or half, or would some not go in there even if they're not virus specific?
I guess based on this, you might expect that some would go in there even if they're not LCMV specific.
Yeah, there's no reason why. I think just because they're proliferating in response to viral antigen that there's so many of them, right?
Right.
You would expect some non-viral ones to go in.
Right. So then, so then they look at the neuropathogenesis of the mice in this figure 5, and we've already talked about the different postures that they see. So, and then in part B, they start to look at the fact that if you just put LCMV in, I guess by the intracerebral route, you're going to get the swelling of the brain and the what they call uncal herniation. So there's this pressure buildup, and so the brain has to go somewhere, so it kind of squirts out to a place where it wouldn't ordinarily be. And they see that same kind of thing happening in these dually infected mice, even though the LCMV is not in the brain. And they see, here's another term, Glenn, this mydriasis, which is just dilation. But every time they say mydriasis, they say dilation, so you don't really have to go look that one up. So, and they have the arrow pointing to the pupil that's more dilated. So that's another piece of evidence that there's some pretty serious brain pathogenesis going on. Let's see.
So this mydriasis happens in LCMV mice infected IC, right?
Right.
Or in the doubly infected where the LCMV is in the IP, right?
Right. Okay. I believe so. Right.
Okay.
So
the point of that is to try and figure out why these mice are getting these neurological symptoms, right?
Right.
So one reason is that there's this increased pressure in the cranium.
Right. And also they're trying to make the point here, going back to reading their subject heading, that the manifestations of CNS disease in the co-infected mice is identical to what they see in mice challenged with LCMV by an intracerebral route.
Right.
So then in the next section, they want to try and get at what you were asking before, is how can they show that the CD8 is what's really involved? So they do CD8 depletion. And the way they do this is they can treat the mice with an antibody to CD8 antigen, and you do it in such a way that it depletes the mice of their CD8 T cells. Is that what they did here? There's that kyphotic posture again.
It looks like it. Because
CD8 depleted animals, right. So...
CD8 depleting antibody, right.
Right. So then they do that CD8 depletion, and then they infect or co-infect the mice or singly infect the mice, and then they plot that all out. And the main thing is that when they have the doubly infected mice that they have depleted, there's a delay in the onset of the death of the mice. Mm-hmm. But they still get death even with the depleted mice. So that leads them to conclude that the CD8s are somehow responsible for at least the timing of it.
So one thing they could have done in addition is to use a genetically modified mouse that doesn't have CD8 cells.
Right.
In which case they might get a more dramatic effect.
Right.
Because the depletion is never 100%. And then that would also allow them to do adoptive transfer, where they could say, okay, we take out genetically all the CD8 cells. We no longer get this neurological disease in the co-infected mice. But then when we add back virus-specific CD8 cells from another mouse that was immunized with, say, LCMV, then we restore disease.
Right.
That's a typically done experiment.
Right. I think, in fact, they allude to doing some of those kinds of experiments in their discussion. But remember, they would have to have CD8 knockout mice that also were transgenic for the measles virus receptor.
Yeah.
And maybe they don't have those, or they're in the process of breeding them, or there's a problem getting those mice. I don't know.
Yeah. No, you're right. It have to be bred, and that might be a problem. Yep.
All right. So then, let's see. What's different? Oh, in part B, they're trying to get at the mechanism of why CD8 is important. And so CD8 T cells, also known as killer T cells, have a number of functional things that they can do. And so they tested one of them, which is the perforin function. So CD8 T cells can release perforin, which makes these holes in the cells that are going to then get killed. And they evidently do have the double mutant mice for that, so they can do that experiment. But what they pretty much conclude is that this phenotype in the doubly infected mice is independent of perforin. So they did the experiment that they could do, but it doesn't appear to be perforin, so it's possibly some other function of T cells, and that's what they're of the CD8 T cells, and that's what they're going to continue to work on.
So this is the experiment that shows that the CD8s are important for that pathology. It's not perforin, it's something else. Could be cytokines released by the T cells, right?
Right.
There's also another granzyme is also involved in cell killing, right?
Mm-hmm. Mm-hmm.
So that could be it.
Mm-hmm.
What would your bet be? What do you think? It's a cytokine?
I'm not sure I would bet on this one.
What would you do next? Say we wanted to find out what aspect of CD8 function is doing this. What would you do?
Well, I think some of the things you described, trying to do the adoptive transfer, or, if there are mice that are not, I think there are mice knocked out for the granzyme, but I think they have a lot of pleomorphic effects, or they're knocked out for a bunch of things. Mm-hmm. Something like that. What would you do?
Well, I might look and see if I could measure levels of CD8 cell cytokines in the brain to get a clue as to which were elevated compared with others.
Right. They did say in data not shown, they looked by RNase protection, so they didn't look at actual protein levels for a whole number of cytokines and chemokines. And they didn't see a significant change between the measles infection alone and the co-infection.
There must be one they didn't look at, because there has to be some difference, right?
You would think.
Unless it's granzyme mediated.
Right.
You know what a young person would do, brand new with their own lab? They would do like a microarray. Right. And do a huge experiment and see what's up in the brains. Right. And then use bioinformatics to sort out the CD8 specific stuff. Mm-hmm. You have to get some clues, because you would turn to genetics next and look at another kind of knockout for a granzyme or another cytokine. But you have to have a clue. You can't just, because these mouse experiments are expensive, you have to cross them with your CD46 transgenics and so forth.
Right. So let's see, this last section, what they want to do. Oh, they're, again, still trying to look at the pathology, and they look at edema. So edema correlates with morbidity in the co-infected mice. So they measure the amount of edema. And so edema is, of course, fluid in a tissue that's accumulated, and it can produce swelling. And, as we discussed earlier, you have this swelling of the brain. It's kind of limited by the skull. That's kind of a bad thing for the brain. And so they show that the mice that are dually infected that have the severe symptoms have a significant increase in the edema that they have in the brain. So they have a significant difference there. But if they deplete the mice of the CD8 positive T cells and then do the dual infection, they don't see that increase of edema. So that says that the CD8s are playing a role in the edema. Then they also look at Evans blue to look at permeability of the blood-brain barrier. So in this LCMV model, what Glenn's lab has shown before is that the breakdown of the blood-brain barrier is probably not the initiating or primary event for the brain pathology. That it does occur, but it's maybe not the leading factor. And so they see what they only call modest permeability of this Evans blue dye crossing into the blood-brain barrier, and it's not statistically significant in this figure.
Edema has to be a consequence of inflammation, right? And some permeabilization of the blood vessel network, because that's how the fluid gets into the tissues, right?
Well, there can be vascular edema, and then there can be
lymph.
No, well, I was going to say cytotoxic, where the fluid just comes directly out of the cells. Mm-hmm. And you can, so it's vasogenic. This is, my student is much more up on this, because what you can do is measure the sodium and potassium levels after you've done the edema measurement and determine which of the two types of edema it is.
I see. So the fact that they don't see any permeabilization of the blood vessels suggests that it might be cellular edema, right?
I think so.
Yeah,
that's one interpretation.
But again, it's a consequence of inflammation, right? So maybe cytokine caused, right? So it gives them some clues, yeah.
So, and then in this last figure, they actually show us some histology and show that, let's see, the middle one is the one that strikes me because they say that they have meningitis and they see more meningitis. I'm not going to be able to find that fact. Oh, wait. The DI moribund mice had more meningitis. The doubly infected moribund mice had more meningitis than the doubly infected mice that had mild disease. So maybe that, the amount of meningitis then therefore is correlating with death. So anyway, in all of these figures, when you see lots and lots of purple stained cells, that's the evidence that you've got an inflammation, an influx of inflammatory cells.
So what we have here, we infect with measles in the brain. So the brain is infected. The cells are making cytokines, right? You've got LCMV in the gut and elsewhere, and you've got these T cells made. And so some of those LCMV T cells and other T cells are drawn to the brain because of the measles infection, right?
Right.
So that is sending out signals which would normally recruit it, but there's no LCMV in the brain. However, those CD8 cells are doing damage somehow.
Right. So, and there's precedence for these kinds of polymicrobial infections where, if you have one virus and you get a second virus, the first virus causes some immunosuppression and makes the second virus more able to establish an infection. They cite some examples of that in discussion. But this seems to be kind of unique in that they're actually seeing this invasion of the CD8 T cells being involved.
One thing that came to mind is mumps infection. When mumps was widespread before vaccination, mumps is quite neuroinvasive, but it's not very neurovirulent. So most kids with mumps would actually, about half of them, I think, would get virus in the brain, but no apparent consequence. So you could imagine that if they had that and then had a second infection elsewhere, they could have had a problem.
Right.
So in cases where some kids got encephalitis or meningitis after mumps, there may have in fact been a second infection that was doing that, similar to this here. And as you said earlier, if you have some of these diseases of unknown etiology, like multiple sclerosis and ALS, maybe a similar thing is going on.
Right.
But it would have to involve an agent in your brain that is not on its own causing overt pathology.
Right.
It's just in combination with something else. So the only one I know of is mumps. You know of any others that might do that?
Oh, I'm thinking of the parasite. There's
Toxoplasma?
Yeah. Toxoplasma, I think, can just be in the normal brain, but if a person is immunosuppressed, then it can be a problem. And I think that's the case. And so you could imagine some kind of trigger that would immunosuppress could lead to a problem, I think.
Yeah.
They also talk about immune responses to other antigens such as tumors or allergens, autoantigens that could contribute and be a factor in some of these kinds of etiologies. So
yeah,
not just viruses.
Right, that's true. This is polymicrobial.
Right.
It's a very cool paper.
Yeah, I liked it a lot. Oh, this is a small point that I made, but throughout the paper, they have a pretty consistent use of their symbols. So whenever they talk about the measles virus, they have a certain kind of symbol, and if it's doubly infected or squares, triangles, it's a minor point, but when authors take the time to do that, I think it makes it easier for the reader. So, I like that.
Yeah, it's often the extra that makes something clearer. That's good.
Right.
All right. That's cool. Hope you enjoyed that. And I think that's about it, right? Anything else before we move on? Okay.
I think that's it.
Yep. Great. All right, the second part of TWiV here, we have had a request some time ago for various people, PIs, early stage, middle stage, late PIs, graduate students, postdocs, to tell us a day in their life, what they do every day. So I thought this would be good because Kathy is new and most people don't know much about her. So we could both do our day in the life.
Right.
See what it's like. You and I are both old people, right?
Right. We were born the same year.
The year
put it that way.
the year DNA structure was determined.
Yep.
So we are both at the same stage in our careers. So it'll be interesting to compare and contrast. But certainly, how you do a day today is very different from when you first started, right?
Oh yeah, definitely. And even so, I moved to Michigan a little over 10 years ago, and I'm not able to do work in the lab as much as before I moved, or even 6 or 7 years ago. It just other things are starting to fill up my day. But
yeah,
that's the way it is. Things change.
So tell us a day in the life of Kathy.
Okay, so I wasn't able to do one day, so I sort of chose 3 days, and I tried to kind of keep track, and so I'll just tell you about these in sort of 30-minute increments unless I say otherwise. And I tried not to just say I did email for an hour, because that could be another way of describing my day. So in the wintertime, I walk into work, and so this first day I'm talking about is February 2nd. It was a day when we had a visiting seminar speaker. But as I was walking in, I listened to the end of TWiV 168, which Rich says is one of his favorites. It's the one called Ode to a Plaque. After I got here, I met with our seminar speaker, who was Lynn Enquist, whose work has been featured on TWiV and who's been on TWiV. So he was our departmental seminar speaker that day, so I got to talk to him for a little over a half hour. Then I spent some time working on a graduate studies issue. I spent some time talking to lab members about what to present in an upcoming joint lab meeting, who should present and what should they talk about. Then I'd been having some computer issues, so I spent some time on that. And about another half hour, my senior student came in. She had some new data that are replicates on experiments for a manuscript, which is nearly finished and ready to submit. So that was nice to see. And then I had some email to do with faculty about upcoming teaching and some graduate studies issues. So I'm the chair of our grad studies program in microbiology and immunology. So I get to do a lot of those kinds of things. So then it was time to go to Lynn's seminar. So I got to have some more real science for the day and hear his talk, which was really great. Then I had some lunch, and then I spent about another half hour sorting out some computer issue. I spent another half hour on some upcoming teaching, and I'm teaching a grant writing class, the breakout sessions thereof. I'm not the primary instructor for the class. Then I had something to do, which was, this is writing a letter of recommendation, and we write letters of recommendation all the time, but this was the first time I ever had to write one for a law school applicant. So that was interesting. I spent about an hour and a half on that draft. And then the next day was one of our days when we were having graduate students, prospective graduate students come visit. So I spent about an hour and a half reading the applications of the specific ones I would meet and some of the other ones coming to visit our department. And then I walked home, which also takes not quite a half hour, and I got to start listening to the next TWiV. I'm still catching up between the 70 and 92, and then I'm kind of current, and I've listened to everything up to 70.
It's funny, when I first told you about TWiV many years ago,
right?
It was in an email, you said, I'll have to figure out how to get it onto my iPod.
Right.
But now you're very good at it, clearly.
Yes. Well, and I also had to figure out when I would do this, which still boggles my mind because I don't know when you have time to do this, but it's great that you do.
So this day, was it all pretty much scripted, or did you was there any randomness involved?
Really, the only scripted thing was the appointment to meet with Lynn and the going to his seminar.
So you basically take things as they come, right?
Right.
Yeah.
Right. Yeah. That's a good point.
It's good. I didn't know. I would have guessed that you had everything scripted.
No.
You seem to be a very organized person, but
no.
Good to know. Because I'm not at all scripted.
Yeah.
Maybe it's a 1953 thing.
Maybe. Okay, so then the next day that I decided to record was the following Monday, in which I had a departmental meeting I had to go to, and also appointments to meet with some lab rotation students. So I walked in, and I was now listening to TWiV 169, because being a Monday, there was a new episode to listen to. And just as I got to the building, Dixon said that Harry Ginsberg had played football for the University of Michigan and had had a hip replacement. And I thought, I don't think he was here at Michigan. So before I even took my coat off, I walked straight to Mike Imperiali's office and I said, did Harry Ginsberg play football here at the University of Michigan? And because Harry Ginsberg was an adenovirologist, Mike was instantly interested too. So we checked it out on a couple of UM football pages, and then Mike later checked out New York Times obituary for Harry. And meanwhile, I had written to Hamish Young, who I guess is his title emeritus in your department?
Yeah,
Must be.
It could be, but he's certainly retired. Yeah.
Yeah. And he's an adenovirologist too. So he said, and we figured out that Harry Ginsberg had been an undergraduate at Duke, so that would most likely be where he played football. And then he did his MD degree at Tulane. And Hamish said Harry did play football and did damage to his knees, which had to be replaced. And he had complications of a serious bacterial infection. So that was my fact checking and what happened as I got into my office.
Interesting.
And then, so that was kind of fun. And then now I've just contradicted myself because I just wrote down here 45 minutes of email. So I don't know what that was about, but okay. Then, so this was after the interview weekend, so we had to look at the evaluations of all the faculty who had met with the prospective students the previous Friday. So they all fill out evaluations online, and I spent 2 hours looking at those for the applicants that we had, and it was actually 2 weekends worth of evaluations. So I spent that time. And then I met with the first of the rotating graduate students. So this was a little bit scripted because I knew I had this meeting in the afternoon that I had to go to evaluate the prospective students. And then I ate lunch, and because it was Monday, I watered my plants. Luckily, I pretty much remember to do that every Monday. And then I went to the graduate admissions meeting for our department, which was about 3 hours in total with the aftermath. The meeting itself was more like 2, but then there was
still long.
Yeah, it is. And then let's see, and that was only for our recommendations to the umbrella or gateway program committee, because then the next day I went to that meeting, which was very brief, so that was nice. Then I met for about half hour with the other rotating graduate student. I then sent out some email about teaching assignments for the grant writing class. I finished the letter of recommendation for the law school applicant. I had gotten comments from a couple of people because it was so new to me. I needed help on, what should I say? And then because it was from a former faculty member, so I had written it pretty much like a promotion and tenure letter, but yet I knew it had to sound like they should be going to law school. So that's why it was a challenge. And then we have hired a new work study student to help in the lab. So I sent email and figured out what we needed to do with respect to that. And then again, I got a little more science as I walked home listening to TWiV.
Wow.
So that's what I like about TWiV, is that on these kinds of days when there's lots of meetings and stuff, I can feel like I'm going to learn a little science on my way in and out.
So it takes you about 2 walks to do a whole TWiV, or maybe more.
Right. Or sometimes I keep doing it when I get home. So, yeah. Then the next day that I decided to do was Wednesday the 8th, which was a teaching day. When I got in, I'm going to be giving a seminar at another university, and they had emailed me and said, please make your travel arrangements and give us your seminar title, and they always want to have your CV and things like that. So that took me about a half hour. Then I did about a half hour of things with graduate student issues, and then another half hour of graduate student recruiting issues. And interspersed with that, I was trying to upgrade my software because the computer is now kind of coming back online and upgraded in a couple of things. I had missed a faculty meeting in January and realized that there was some important financial model document that I wanted to know about. So I spent about 10 minutes looking at that. Then I got to do a little more of my own lab science. My senior student brought in the draft of her manuscript, and I started looking at that. Had another half hour consulting about graduate admissions. And then I went to the class. I must have eaten lunch in there before class. And then I went to the grant writing class, which is, for the moment, the first couple classes are an hour and a half, and then soon they'll go to a 2-hour format. But it was about 2 hours total because there was some aftermath. And then I spent about an hour with my students' manuscript draft again. And then I had the assignments that the students had turned in the grant writing class. So I spent about 2 hours looking at that. And then, that day I also listened to TWiV on the way in and on the way home. So cool. That's more science. So I did have somebody ask me once, and I always remember this, you're a microbiologist. So does that mean you look under the microscope all day? And I just laughed. And, I love it when somebody says, can you come look at my cells? Or is this time to harvest? Yeah. It's because then you do get to go look under the microscope.
Yeah, but these 3 days you didn't.
Nope.
Yeah.
So how about you?
Well, I wrote down just a bunch of things that I do. Try to remember, not a daily thing, and when I first came here as a new assistant professor, I was in the lab a lot. Not all the time, but often most of the day doing experiments right next to techs or students and eventually postdocs. And as time went on, I did less and less than that. Yeah. I try to still get in the lab and do things. I split my HeLa cells 3 times a week, so we have a spinner of HeLas, and I do that Monday, Wednesday, and Friday. So it only takes half hour, but it gets me in there. Sometimes there are people in there. I usually do it first thing in the morning. As recently as a couple of years ago, I was actually doing some experiments. We were doing a project on zinc inhibition of rhinoviruses, but I just couldn't continue it anymore, and actually one of my students has now taken it on. Okay. I do quite a bit of teaching. I teach medical students here. I teach graduate students. I also teach an undergraduate virology course in the spring, which is what we're doing now. So I spend time making exams, preparing lectures,
answering emails.
It takes a lot of time for the course. The whole course I teach myself with so it's 26 lectures, and 4 lectures are given by Saul Silverstein. Wow. So it's quite a bit of work. I have to go downtown to do that. So it's basically I leave here at 3:30 and I come back at 6. Wow. So it's a big chunk of the day, plus preparations. Yeah. It's a lot, but I like it.
And that's 3 days a week or 2 days a week?
2 days a week, yeah. And then I often teach various things around the city, so there's always some teaching. I go to seminars a lot. We have a department seminar once a week and also a student seminar once a week. And every other department here has seminars, so I could spend a lot of time going to seminars, but I really try and limit them. I meet with people in my lab. I have a standing appointment every week with everyone to talk to them, but then people wander in all the time. Anyway, we have a lab meeting every other week, which we do jointly with an immunology lab here.
Oh, good. So all the immunology wasn't too foreign to you.
No, so we are actually collaborating with Kang Liu, who is a dendritic cell immunologist.
Cool.
She discovered a subset of DCs that are in the brain. So I said, well, why don't you look at a virus that infects the brain? And she's doing that. So we're using encephalomyocarditis virus, and she finds that it increases these DC populations. So I'm learning all about the markers you use to identify different DC populations. So we have a joint lab meeting, and it's really nice.
Yeah, that's great.
When I eat lunch, it's usually with someone here, either Saul Silverstein or Steve Goff or whoever else wanders in. We just eat right here in the office. People are always wandering in to talk, 10, 15 minutes at a time. They sit down, we chat about science usually, or complain about H5N1 or XMRV or whatever the most recent issue is. I travel quite a bit, either to a meeting, to give a seminar, to do a road TWiV. So every month I'm usually somewhere for a couple of days, study section. Spend a lot of time doing email. But the offshoot is I don't get letters anymore, so that's good. We have a department office downstairs. I hardly ever go there.
Right.
I used to go every day to get mail, but there's no point anymore.
Right.
I read a lot of papers about our work, but also looking for topics for all the podcasts. It makes it kind of fun because I get to read a broad array of papers. I review papers for various journals. I review grant applications. I write grant applications. And lately I've been talking to journalists a lot, either print journalists or broadcasts. Like yesterday I had 2 different people I spoke with because, again, the continuing H5N1 story, there's a lot of interest. And once your name gets out there, people call you all the time. I have had just inquiries everywhere. Yesterday, one of them was from a writer from USA Today. So I think it's important to talk to these individuals. I never used to, but I decided at some point that it was important. So I do quite a bit of that. And next week I'm going to go record a 1-hour radio show somewhere here in New York City, and there's always something. So it does take time.
Yep.
Mostly I try to do the podcast stuff at home at night and on weekends, but sometimes, I see an article here, I put it on the agenda, or I correspond with people about it. I say, hey, can you do TWiV on this day or whatever? So there's always some bleeding. There's no absolutes. But that we do record all 3 podcasts, TWiV, TWIP, and TWIM right here. And so that takes, for TWiV, sometimes 2.5 hours.
Right.
It's like now we are doing TWiV on a Friday afternoon, usually it's from 2 p.m. to 4-ish or so. So that is dedicated to the podcasting, but I think it's worthwhile.
Oh, yeah.
So that's sort of the mix of things that I do. I like it. I don't think I would need to change it. If I want, I would love to do lab work, but that would take away from a lot of other things because you really need to be dedicated with lab work to follow it up. And I'm not sure that my contribution would be so useful that it's worth cutting something else out. So I'm happy with the composition. What about you? Are you happy with the way things are? Would you change something?
If I could work more in the lab, I think I would like it.
Yeah.
Or if I could figure out more time to read papers, I would like that. But I used to work in the lab quite a bit, and I had, my thesis advisor, John Holland, worked in the lab the whole time I was a graduate student, and he, I think, and he had a lot of teaching too. But I think what's done it for me is the grant writing pressure, and then that sort of took me out of the lab, and what I could do. Things like I could go in and clone something because you can always stick cloning in the freezer or, and wait for the next step. But the kinds of experiments we do now aren't those kinds of experiments. So it's not so amenable to just dropping in and doing something. So when I do have time, I'd like to say that I can help people. I'm still the only person who knows how to cesium chloride purify the virus. So whenever somebody's project requires that, then I'm in there showing them how to drip the gradients and all that kind of stuff. But
one more thing I wanted to ask you. You must take some work home, right?
I do. But it also tends to be sometimes that you feel virtuous taking the work home, and then you get home and you think, I need to have a balanced life. I need to eat a decent meal or do something, and then you feel kind of like, well, here I am dragging this thing back in that I brought home and didn't read, but at least I brought it home in case I had the opportunity to read it. So. Yeah. I tend to work, longer days, and then kind of when I'm home. So, but it depends. When I'm teaching, then I'm up late at night and early in the morning reading whatever it is for class, that sort of thing at home.
Yeah, I'm increasingly conflicted. We have 3 kids, and they need time, and I have a long commute. It takes me at least 2.5 hours a day. Yeah. So I get home, I often want to work, but I help my kids do their homework, and then they go to bed, so I get some more work done. On Sundays is TWiV or TWIP or TWIM editing day, so it's hard. Yeah, so it's always a balancing act, and I could spend probably every night and every hour on the weekend doing work-related stuff or podcast-related stuff. But as you said, you have to do something else, and especially if you have kids, you have to pay attention to them. Otherwise, you shouldn't have had them in the first place.
Right.
It's not fair. So it's a balancing act. It's always juggling, and you're never going to get the perfect combination, but you do your best.
Right.
Cool. Well, I hope that's a good sense for everyone. We are senior PIs. This is what we do. We'll get a middle career and a young career PI in some time to do this, and so you can get a sense. I have my student is coming on to TWiV next Friday, maybe.
Ah, yeah.
She could tell us what she does.
Yeah.
As well. All right. Let's do some email here. The first one is from Oystein. I believe that's how you pronounce it.
Yeah, I don't know.
O with a slash through it, Ystein. Dear Vincent, I'm writing to thank you for a truly inspiring present. I'm a Norwegian veterinarian doing a PhD in virology at the Norwegian School of Veterinary Science. I'm a big fan of your podcast, and a little while ago, my girlfriend apparently contacted you regarding our 3-year anniversary and my birthday in December. I didn't know if you remember this, but she asked if you could write a little letter to me for this occasion. To my surprise, included in her gift was a envelope marked Columbia University, New York. No technological gadget or other materialistic gift could have beaten this present. Inspiration can't be bought, and I really appreciated that you took the time to write me. You and your crew on TWiV are truly a source of knowledge and motivation. Your podcast has really educated me both in virology and the scientific way of thinking. So his girlfriend had contacted me. She said, could you just write him a letter for his birthday? He loves TWiV. I said, sure.
Great. So what did you say?
I said, hey, Ystein, how's it going? This is Vincent, and we're getting ready to do a TWiV. I don't know, I just wrote a couple of paragraphs of what I was thinking about, and I said, how's your work going? Congratulations. And, I FedExed it, and was happy.
Yeah.
Someone else has also recently asked me to do this, and if you're listening, please email me because I forgot, and I can't find the email. Someone said they're getting married, and they want to read a letter at their wedding or something. So I'm happy to do it, but you got to remind me because I can't find the email. I searched for wedding and all this other stuff. I can't find it. Anyway, Ystein continues, I'm in my PhD. I'm working on a novel reovirus called piscine retrovirus, PRV. Reovirus, PRV. PRV is associated with heart and skeletal muscle inflammation that is an emerging disease in farmed Atlantic salmon. The virus was actually discovered in 2010 in collaboration between the lab of my supervisor, Espen Rimstad, at the Norwegian School of Veterinary Science and Ian Lipkin's lab at Columbia, using high-throughput sequencing. Thanks to their work, a PhD position later opened up to study this new virus, and that's how I got into virology. I really enjoy the field, and TWiV has made it even more interesting. Thank you, Alan, Rich, and Dixon, for your superior podcast. Keep up the good work. I hope you are motivated to continue for a long time. By the way, the temperature in Oslo is negative 5 degrees centigrade.
Except it should be Celsius.
Celsius. I got to remember that. How am I going to remember it?
I don't know. Just,
I got to put a sticky with Celsius right on the monitor. That'll do it. And then, the next time I record, I'm going to look at it and go, what the hell is that for? Because I'm so scattered. Yeah, we are motivated to continue a long time. I'm happy to do it as long as I can get people to do it with me. Mm-hmm. I'm lucky that Rich and Alan and Dixon are happy to do it, and all the guests. Mm-hmm. But if no one wants to do it, I can't do it myself.
No, you put in a lot of time for this.
But I think I could get people always, don't you think?
Oh, absolutely. Yeah. Yeah. Everybody wants a star turn, their 15 minutes of fame or whatever. Yeah.
Right.
So,
Kathy, you want to take the next one?
Sure. Thomas writes, firstly, Happy New Year to the entire gang. I look forward to another wonderful year for all 3 podcasts. I hope you reached your 1,000 mark for your listener survey, and I'm excited to see where TWiV goes from here. Did you get to 1,000?
Yeah, we did actually just a few weeks ago.
Oh, good.
And so it's great. We have a great survey of who our listeners are, and now we are trying to get some advertising support. Mm-hmm. Yep. So thanks to everyone for doing that. Did you fill it out, Kathy?
I did.
Thanks.
I did.
Good.
So Thomas continues, my main reason for writing today is in response to some comments made during the year in review TWiV number 164. I believe it was Rich that made the observation that most of the articles that were chosen were somehow implicated in human disease and pathogenesis. He then went on to mention that many of the issues with some of the more controversial stories dealt with the public's knowledge of science and how projects, such as the H5N1 influenza project, are portrayed in the media. P.S. Thank you, Vincent, for the articles on the TWiV Facebook page. It helps me to keep on top of the issue without having to search through various sources. The P.S. is right in the middle of the letter. It's a little confusing, but okay. So now, I'm not sure what your experiences in college were, but mine involved that of a small Catholic liberal arts college in rural New York State. Here we were required to complete the compulsory courses to obtain our BAs, BSs, etc., along with the compulsory courses as part of the liberal arts curriculum. Topics in this curriculum included microeconomics, sociology, sacred texts of various religions, and an intro to science and the scientific process, to name a few. I felt I left the school with a general understanding of many topics to an extent where I am less intimidated by balancing a checkbook, confident in being able to converse and interact with peoples of various religious backgrounds, and obviously an extensive knowledge of science given my studies in biochemistry. My question to you, gentlemen, is this. Do you feel that a liberal arts education would help the situation discussed during TWiV, where it appears that the general public is just not competent, in quotes, in the scientific process and how science actually works? I feel that my introduction to foreign subjects such as microeconomics and religious texts was sufficient for me to feel less ignorant of the material and confident enough to make prudent decisions should I be faced with one concerning such topics. Thus, I would hope that my classmates who are not science majors and were introduced to a very basic level of scientific concepts, to very basic level scientific concepts and the scientific process would similarly feel more confident in not being led down the wrong path by media or other sources. So what do you think, Vincent?
I don't know if liberal arts is enough because I think the amount of science in that is pretty minimal, and I think most people forget it soon after they graduate from college.
Yeah, I suppose it depends how much they're required. If it's maybe just 2 semesters of science, that might not be enough.
And then you get into the problem of if you want them to take more, then it takes away from their major. And
right.
So here at Columbia, we have what's called a core curriculum for all majors, which includes science, but it's not very much. Mm-hmm. It's biology, chemistry, a little physics, but it's especially beginning courses for those individuals, and they're not major courses. So I don't think it would help you understand H5N1 or XMRV in the end. Now, everyone can't be a science major.
Right.
So I think the solution is to listen to TWiV.
There you go.
For viruses, anyway.
Right.
I think you have to go beyond what you get in school. I mean, a good liberal arts education will give you the tools to be able to figure out things and understand, but you have to go beyond it. Maybe though the solution is before that, to give younger kids a better science education. That could be done in middle and in high schools, right?
I don't know if they're also time constrained in how much science they can get, but maybe it's also a matter of not just quantity but quality. So I think a liberal arts education is certainly a good start. It's better than not having any science classes. And the listeners on TWiV span a wide range of educational backgrounds, and so you're right. Listening to TWiV can be a way to further scientific knowledge, but it also shows that they're out there trying to learn different things, and so there's at least some population that's been able to do it with whatever education they've had.
The people who listen to TWiV want to, they're interested and they're motivated, but there are many people who don't care, but should understand viruses to be able to make decisions and understand why we vaccinate, and
Right.
But many people don't want to do it, and you can't have everyone listening. I understand that.
Right.
Now, Thomas is not a science major, right? Is he?
It sounds like he was science, although he doesn't.
Yeah, maybe.
Didn't really say.
So Thomas listens to TWiV, and he probably understands a good part of it. But we were trying to get he studied biochemistry, right? Okay. We have to get the people who don't have a good science background to understand scientific issues, because really society is mainly driven by science and technology issues these days, right? Okay. I mean, there's so much that everything we do revolves around science and technology, from our health to our ability to work. So I think it's really important to understand them, particularly when fear pops in, when it's fear of a pandemic or something like that. Right.
Lack of knowledge can lead to fear.
So you think a liberal arts education is a start, but you have to go beyond that? Is that what you're...
I think we all need to continually educate ourselves on things. And unlike Thomas, I didn't have a microeconomics class, but yet I feel like I do know some things about the business world, and I read the business section in the newspaper and things like that. But there's only so much time that we can learn all these things. So.
It's interesting you say that you read the business section. So I think a lot of people get their science from newspapers and television and radio, right? Mm-hmm. And while there are so many good science writers, as you know, there are also less than good ones, or ones that make assumptions that aren't correct. And so I would urge people not to just do that, to go beyond science writing in the media and go to podcasting, blogs, or even picking up a book and reading about science yourself, or take a course. If you have time and you can take a course at a college, that's another way to do it. Mm-hmm. But I personally would read blogs and listen to podcasts because I love consuming that way. Mm-hmm.
Yeah, I think you shouldn't limit yourself to any one source of information. So, while I read the business pages, I'm not going to take everything there at face value, because those stories that I know something about firsthand that I've seen in the newspaper, they don't always get portrayed the way that, that seemed like that was reality. So knowing that is a good thing, I think. So, Tom, do you want to... I was just going on. Finish. Thomas writes, I apologize for the length of the email, but I sincerely believe that the liberal arts education I received was excellent for the reasons I alluded to above, and I wonder what your opinions are on the matter. Keep up the great work, and all the best in 2012 and many years thereafter.
So listeners, what do you think? Let us know. Send an email. What do you think? You have a liberal arts education? Does it help? Does it hurt? Does it do anything? Let us know. It's an interesting question. Next one is from Mike, who writes, hello, TWiVites. I think TWiVites is a new one. I just recently listened to episode 164, was delighted to hear your response to my question. Thank you so much for taking the time to contact your colleague and relay my question to her. That was Kathy Collins, your colleague over there. I very much appreciated her taking the time to respond in the depth of her answer. However, the answer left me with yet still more questions. It happens.
Yep, that's good.
The colleague you mentioned in your response seems to be pursuing a type of therapy that involves using various cellular signaling factors to activate transcription of the viral genome in an otherwise latent cell. Once the viral genes are transcribed and the virus begins to actively replicate, antiretroviral therapy is used to prevent the spread of the virus to uninfected cells, while attempting to kill the viral infected cell with another form of antiviral therapy. Your colleague, Kathleen Collins, said that the trick to all this is finding a balance between efficacy and toxicity. In other words, kill the bad cells but leave the good ones alone. I found her answer to be very complete and satisfying to the question that I asked, but now I have a few more. One, is it true that HIV can only infect cells that are positive for the CD4 receptor? Kathy Collins says, almost always there are reports of cases in which HIVs have evolved that can infect CD4 negative cells. This is the exception rather than the rule. His question number 2, if question 1 is true, does a CD4 positive cell infected with HIV express any cellular surface markers that uniquely identify it while not alerting the immune system when it is still in a latent state? And Kathy says, for latently infected resting memory cells and hematopoietic precursor cells, the cells do not express the necessary transcription factors required for efficient transcription of viral gene products. Therefore, no viral proteins are expressed. It would be great if there was a cell surface marker for these cells, but to my knowledge, there is not. So then he goes on to say, if it were true that viral proteins were expressed, you could engineer an antibody and then get rid of the cells that way, but there aren't such cell surface proteins, so no go. I recently saw a program on the Discovery Channel that showed a recent experiment involving rats and longevity. In this experiment, the researchers used this kind of antibody tagging system to remove senescent cells from the bodies of aging rats. The whole process used a piece of equipment that looked somewhat similar to a dialysis machine. The machine used a magnetic field to pull senescent cells out of the blood as the blood was filtered through by pulling on a ferromagnetic antibody attached to those cells. The blood was then pumped back into the rat. The rat behaved like a much younger rat after that and did not show many of the aging-related diseases that other rats who had not undergone this procedure did. I suppose my ultimate question is this: could the same process be adapted for HIV or for any viral infection for that matter where a unique surface protein, glycoprotein, or any other kind of marker is expressed on the cellular surface? Well, the answer is sure, and we in fact use antibody therapy for similar reasons, to get rid of certain types of cells with specific markers. And you don't have to pull them out by magnetic approaches. The antibodies will help destroy the cells by a variety of mechanisms. So it's completely doable, and there are monoclonal antibodies that are used for a variety of conditions. One I'm thinking of right now is a monoclonal antibody against a B cell marker to get rid of B cells in people who have excessive antibody responses, for example.
And it's parallel to what we had in the paper today where they were doing the CD8 T cell depletion.
Yeah, exactly. Yeah, you just add antibody, enough of it, and it will clear the cells out. You don't have to use magnetic beads. Thank you for taking the time to read this, and I will understand if you do not respond due to the length. Now, it's no problem. Also, I would like to thank you for what you do. I listen to your podcast, Twim, Twip, and Twiv, on my way to work, and it makes the drive go much faster. Please forgive me if this is an ignorant line of questioning. I'm somewhat of a layman. I have only a couple of bachelor's degrees in science. Once again, thank you. You don't sound like a layman. A couple of bachelor's degrees?
Yeah.
P.S. Please tell Dixon that I live in the Chicagoland area and will be touring his vertical farm project in the next few weeks. Well, Dixon's not here. I'm sorry, but I'll tell him.
What I like about this is that it shows real creativity. You know, he saw this program on Discovery and he was thinking about how they are pulling out these senescent cells and how that changed the rats. And then he made the extension to doing it for a virus. I like that.
We have great listener emails, I have to tell you. I think it's the best of any podcast I've listened to. We're really lucky.
Yeah.
All right, Kathy, take the next one.
Okay, Eric writes, Dear professors, I am a TWiV listener and I enjoy your podcast and trust your judgment. I recently came across this story. I would appreciate your thoughts. Is there any truth here at all? The story is fairly short. Knowing what the government did to soldiers in the 1950s, I can't reject this out of hand. If this is all just BS, then it needs to be exposed. Regards, Eric. So then he provides a link and then a long story. And the gist of it is that it's talking about whether there's a link between cancer and the polio vaccine. And this stems from the fact that in the polio vaccine, the Salk vaccine, it was subsequently found many years later that there was SV40 virus. So that's true. And I think there's even maybe evidence that there might have been... No, that's all. Okay. So that's true. But then the rest of this article, as I told Vincent earlier, I couldn't even bring myself to read it because it had the sense to me of just putting together a bunch of facts or facts sounding like things, but that I didn't have a way to really document. And so it reminds me of some of those kind of internet stories that you get and that I like to go and check out on Snopes.com, except that actually I didn't go check whether this one is on Snopes.com or not. Maybe some other experts have already answered it. But it takes something for which there's some factual information and then goes on and makes theories and quotes names of famous people. And so it, I don't know. What do you want to say, Vincent?
So, I mean, the SV40 was certainly present in polio vaccines, was given to a number of people, and many studies have shown that this does not cause cancer in people. In fact, Pat Moore, who was on TWiV not too long ago, we asked him, he said, no, there's just no evidence that SV40 given to people caused any tumors. There are a few statements, well, there are many statements in here that are just not wrong. For example, the foremost virologists studying AIDS, Robert Gallo and Luc Montagnier, agree that it is a virus from the African green monkey that causes AIDS. This is not true. Okay, the precursor of AIDS came from a chimp or an ape back in the early 1900s, not from an African green monkey. They have designated this virus HIV for human immunodeficiency virus. Another virus carried by African green monkey is SV40. And then they go on to say that HIV and SV40 cause similar symptoms.
Yeah, not true.
Which is not true. And so they're trying to make a parallel between SV40 and HIV, and that it's been put into people, and we're telling people to get vaccinated, and we're giving them these cancer-causing viruses. So this is just one false fact linked to another. You know, there are a few things that are correct in here, like, yes, the polio vaccine did have SV40 in it, but we are not unleashing a plague of biblical proportions, as the last sentence of this article says.
Right.
So no, SV40 doesn't cause tumors in people. There's no there's no conspiracy to give people contaminated vaccines. We are just trying to prevent illness, and I don't know why people have to believe there's a conspiracy or malfeasance or any of that sort of thing. So this is all wrong pretty much in its conclusions,
right.
Eric.
But thanks for checking.
Yeah, I mean, I'm glad you trust our judgment. But there's really nothing to this at all. It's a typical non-fact sort of article linking a few wrong things one after another. It sort of sounds compelling, but it's not right. Okay, let us do one more here. This is from Margot, who writes, I've just started listening to you guys, no gals, and am enjoying it. Well, we got a gal today.
Yep.
We'll try and get more.
Yep.
We try.
Yeah, and you've had some in the past, too.
Yeah.
Kathy Collins, Michelle Osborne, others.
Yep.
Yep.
Here's my question, and I've only listened to 2 so far. Maybe you've addressed this already. Do viruses have any positive effects on humans? We've discovered so many important roles bacteria play, but the only good thing I've heard about viruses is that they have influenced our evolution. It's good that you've heard about these beneficial bacteria. Not everyone understands that. Yeah, we have talked a lot on TWiV about the theory that viruses may be good, but I don't know any human evidence, direct evidence. And we often cite a paper from Skip Virgin's lab. It's called Herpes Virus Latency Confers a Symbiotic Protection from Bacterial Infection. And this is in a mouse model. He shows that infection with a herpes virus protects against Listeria and Yersinia, which are, of course, bacterial pathogens. And so you can't, of course, extrapolate to people from that, but the precedent is there. So I think we're still in early days, but eventually we will find some positive evidence in people. It's not easy to do. What do you think, Kathy?
Well, this reminded me of another woman that you've had on TWiV, and that's Marilyn Rusink at the first TWiV that I experienced, which was at the ASV in Bozeman. And so she's written a review article that's unfortunately in Nature Reviews Microbiology, so it's behind a paywall, but she has a table in here of possible, the title of the review article is The Good Viruses, Viral Mutualistic Symbioses. And she mentions the Barton and Skip Virgin paper that you mentioned. And it's true. I don't think there's much evidence yet in people. That's there's a link about here that herpes viruses suppress HIV infection. Mm-hmm. So that's somewhere in the paper. But so that's a different one from the Barton and Virgin paper. But she's got lots of examples in plants, bacteria, fungi, and the parasitoid wasps, which I know has been talked about on TWiV before. So, in fact, I don't know if it was that episode or subsequently you thought that maybe you shouldn't use the tagline about the kind that make you sick.
That's
because
right.
not all viruses make you sick. They might be good for you. So,
yeah, I can't get away from the tagline. It's too good.
It is.
Yeah, given that there's so many beneficial viruses in plants and wasps and so forth, there must be in humans. We just haven't found them yet.
Right.
And so I don't see why it would be restricted to everything else. Yeah, that's a great review, by the way. Mm-hmm. We'll put a link to that. Yeah, and Marilyn gave a talk. Actually, she was on TWiV, and she made the point that viruses aren't all bad, and gave us examples. And her favorite one is the grass that grows near hot springs that is able to survive the temperature because it has a fungus in it, which in turn has a virus. And if you take the virus out of the fungus, the grass can't grow under high temperatures. So it requires both. It's really cool.
That is cool. Yeah.
All right. Let us do a couple of picks of the week. What do you have for us, Kathy?
Okay, so I have something that was in Science January 27th. So Bruce Alberts wrote an editorial about teaching real science. And the issue has the first of a series of what are called IBI series winners. And these are essays about science curriculum modules for colleges, particularly for non-majors. And it's kind of an active learning experience where the students start to work out things and then design experiments themselves that are open-ended and trying to get away from the canned cookbook labs that you might have in a course. And in his editorial, he also talks about a science lesson for kindergarten students, children age 5, and he spends a good bit of the editorial talking about that. And so the one that's in this particular issue is about light, sight, and rainbows. And then I checked today's Science because it's the last Friday in February, and the second IBI series winner is there, and it's about phenology, which I didn't know about, or maybe it's phenology, the study of recurrent natural events, such as cyclical or seasonal changes. And this article describes using campus trees to look at this, and it's from our neighbor, Michigan State University, because they do have a really beautiful campus with lots of trees. So that's my pick.
So this is mainly college level?
These essays are college level, and the first year it was just about science, and for this next year they've expanded it. They'll do engineering. And then, as I said in Bruce Albert's editorial, he talks about the U.S. National Science Teachers Association, which evidently is a forum for these kinds of things for younger students.
So these would be mostly experienced by science majors, I guess, right?
Well, I think it's supposed to be for non-majors, or I somehow got that somewhere, so that they can experience what it's like to formulate a hypothesis, figure out a way to test it. So they have a picture, for instance, of solar ovens, and one is black and one is silver, and they figured out that the black one got a higher temperature, but they were able to bake cookies in both of them. So
that really goes back to whose question was it? Thomas in liberal arts education. If you included this, because I think that's the big problem in not understanding science, because you never do real experiments. Right. And you never understand what it is to make a hypothesis and test it and then go back and readdress it. And if this could be built into curriculum in a meaningful manner, not in just regular labs, I think that would be great, right? I think so too. It would help a non-science major really understand what's going on. I wonder if Alberts likes what we do here on TWiV. I don't know. Does he even know about it? Probably not, but if he did, do you think he might? Oh, I would think so. I think anyway, AAAS's mission and NSF's mission and so forth, they're trying to educate not just scientists about science, but everybody. Yeah. So I think TWiV and TWIP are a way to do that. They should do an article on the use of podcasting in education, in science education, for example. It's a good idea. Raise awareness. My pick is a graphic that I saw a while ago, and it is called The Long and Winding Road to a PhD. Have you seen this before? No. It is a graphic, and actually there are a number of these around. I think we've put some other ones up as pics before. It's from onlinePhDprograms.net. Okay. In the U.S., there are more than 20 million students enrolled in graduate school. For many, the appeal of the life of the mind, being buried in books and surrounded by the intellectual elite, is the ultimate fantasy. But many students setting foot on their new campuses for the first time are unaware of the severe drawbacks that await them, from the huge costs to the massive time investment and dismal job prospects. The road to a PhD is long, winding, and full of pitfalls, something the establishment would prefer you didn't know about. It goes through every year of graduate school and gives you numbers. Like a Candyland game board or something. Who drops out, and in the end, what your job prospects are. And then, of course, at the end, nearly half of science and engineering PhDs do postdocs. They pay an average of $37,000 a year, take 3 or more years, and are often required for tenure-track positions in the sciences. They go over the two-body problem. The earnings, PhDs command only a 3% earnings premium over master's degrees across all disciplines. Wow. So lots of statistics. Yeah. It's kind of nice, so check it out. I like that. I'm not sure it's true for all PhD programs, but it's an overview. Right. We also have a listener pick from Greg, who writes, Dear TWiV, the epidemiology episode with Michael Walsh was great. I loved the philosophical detour into counterfactual statements, time travel, and the meaning of causation. TWiV may indeed be viral, but from listening to it, I feel inoculated against the micro-specialization that is endemic to so much of science. I love that. I think that's great. Dr. Walsh's rigorous stance against making statements of causation based on epidemiological studies reminded me of the following XKCD webcomic, and that is his pick. That's great.
That's a good one, yeah. I sent that to one of my colleagues. He said, that's better than the one I use in my statistics class.
Is 2 stick figures. I used to think correlation implied causation. Then I took a statistics class. Now I don't. And the other person says, sounds like the class helps. And he says, well, maybe. Yeah. Wonderful. Allow me to thank you all for putting on TWiV, TWIP, and TWIM. I'm a physicist who has inexplicably become a postdoc in biomedical engineering. I find each of your podcasts to be a revelatory journey into biology. Often I return with something I feel like talking about to complete strangers.
Wow.
I think that is another. This guy has 2 great sentences in this email.
Yeah.
That's it. Isn't it cool when you find something that you're really amazed about, you want to just tell people about it.
Yeah.
So I'm so glad that TWiV makes him do that. That's great. Yeah. Thank you, Greg. That will do it for TWiV 172. You can find us, of course, at iTunes, the Zune Marketplace, TWiV.tv, and microbeworld.org. We're all over the place. And we do have an app for your smartphone or Android device at microbeworld.org. If you are new to TWiV and you like us, you can help us out by leaving a review over at iTunes. That helps to keep us visible. The more reviews a podcast has, the more likely it is to stay on the front page of the directory there, which is how more people discover us. We love getting your questions and comments. Of course, send them to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan. Thanks for joining us, Kathy.
Thanks, Vincent. It was my pleasure. This is a lot of fun.
It's a little different from the on-the-road type TWiVs, isn't it?
Yep, and it got me to do something different from my usual day in the life. This isn't usually in my day.
Yeah,
so.
Well, you end up learning quite a bit.
Definitely.
That's what I find. You should come back again with the entire crew and experience the Condit and Dove of TWiV.
Oh, yeah. I've experienced it just listening, so yeah.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.